Good morning, students. Today we will start with blastulation. In the last class, I have told about the types of eggs. Okay, so that is based on the yolk, presence of yolk, and the amount of yolk. Today we will see about blastulation. So coming to blastulation, so blastula, what is blastula? So here blastula is the first stage in development of any organism. If you take any organism after the fertilization, so when the sperm and the egg will get fertilized, zygote will be formed. After the zygote, it will start to cleave. After the cleavage, the formation of the cluster of cells is called as the blastula. Therefore, the blastula is the first stage in development of organism. So, blastula is a Greek word. Blastos means sprout. So, it is a hollow sphere of cells. Okay, so this is made up by cells. Those cells are called as blastomeres. So a blastomeres will be surrounded by the central cavity. The cavity will be filled with fluid and this cavity is called blastocyst. So here blastula will be formed during the early stage of development. So here blastula if we will compare in different organisms, so it will be a different. Okay, coming to introduction about the blastulation. So there is a difference between blastula and blastulation. As I said, blastula is the first stage of development, but blastulation is the process of formation of blastula. The process of formation of blastula is called blastulation. Coming to introduction, as I said, the embryo development begins after the fertilization. When the sperm will be fertilized, then egg to become zygote, then it will undergo cleavages to develop into a ball of cells. So initially it is called as morula. Morula is nothing but a cluster of cells. So once the morula will start to form the cavity at the center, this is the blastulation, otherwise the forming the blastula. So only when blastocyst is formed in the embryo, then it will become blastula. So here coming to different types of blastula. So today we will be discussing about introduction. I discuss what is blastula, what is blastulation, types of blastula and what is blastocyst. So there is a difference between blastulation and blastocyst. Okay, so here coming to blastocyst, as I said, what is blastula, what is blastulation? Coming to blastocyst, blastocyst is a cavity in the blastula which is filled with fluid. This fluid will have the mucopolysaccharides. Next, coming to the types of blastula. So, as I said, the different organisms will have the different types of blastula. So, there are coeloblastula, stereoblastula, periblastula, otherwise it is also called as superficial blastula, discoblastula and blastocyst. First, coming to coeloblastula. Coeloblastula, it is a hollow sphere and blastocyst is filled with mucopolysaccharides and blastoderm is a single layer of cells, only one layer of cells you can see. Okay, so such blastula we can observe in echinoderms. Okay, so in echinoderms, the blastula is called as a coeloblastula because it is a hollow sphere and a blastocyst will be at the center of the blastula and it will be surrounded by a single layer of cells. So this type of blastula is called coeloblastula. Next, coming to the superficial blastula. So here superficial blastula is also called as periblastula. So here some of the insects. So here they have got the only microlecital means small amount of yolk. So in such type it is the blastulation will take place at the periphery. So therefore it is called as the periblastula. Otherwise, it is also called as the superficial blastula because the cleavage will be a superficial layer. There is no blastocyst in it. All the nucleus will divide and they will move into the periphery of the zygote. Okay, so therefore it is called as the periblastula because at the center it will be filled with small amount of yolk. The cavity, the blastocyst will be filled with yolk. Therefore, there is no blastocyst. So superficial blastula, otherwise periblastula. Example, insects. Next, coming to the stereoblastula. So you see stereoblastula, it is a solid blastula because there is no blastocyst cavity itself. Therefore, it is a solid blastula. So this type of mainly due to the spiral cleavage we can see in molluscan. Molluscan egg undergoes spiral cleavage. Therefore, there is no blastocyst. Such blastula is called as pseudoblastula. Next, coming to the discoblastula. So here discoblastula, it is quite common in bird egg because here it is a macrolecythal. In birds, it is a macrolecythal means more amount of yolk. Therefore, the whole blastula, no? So this will be towards the animal pole. Here, blastula we can say only towards the animal pole because towards the vegetal pole there is a large amount of yolk. Therefore, it will not undergo any cleavage. So here blastula is only restricted to the animal pole. Therefore, it is called as the discoblastula, and also it will appear like a disc at the animal pole. No? In the egg it will appear like a disc, and also it is very small blastula with multi-layered of cells, flattened disc shaped. Okay, so here multi-layer cells will be separated. In between there will be a narrow cavity which will become blastocyst. So this type of blastula is called discoblastula. Next, coming to the blastocyst. So here in the mammals, you know that in the mammals, most of the human being and it is the elliptical egg without any yolk. Okay, so this type of blastula is formed in mammals by regular cleavage. So here the small cavity called blastocyst will be a very smaller, and it will have the cluster of cells. It will be differentiated into distinct groups, like we'll call it as a trophoblast cells, otherwise the nutritive or the formative cells. These are the types of blastula in different organisms. Next, we will move to blastulation in sea urchin. As I told in the last class, the sea urchin is a lower invertebrates. So your egg is microlecythal, only a small amount of yolk will be there, and fertilization is external. Outside the body, fertilization will take place and zygote will be formed. So then the blastula will form after cleavage. So here in sea urchin, after 28 cell stage, the blastulation will start to form. Okay, so once the cells will become 128, then the blastulation will start. Means the central cavity blastocyst will start to produce. So blastulation will be starting from 128 cell stage. We'll see the cleavage. This is what picture I am showing is the sea urchin. Okay, before going to the blastula, this we will see the cleavage. So here first coming to the cleavage. So here the first cleavage in sea urchin, it will exhibit the radial holoblastic cleavage. In cleavage also we have the two types, that is the complete cleavage and the incomplete cleavage. Where there is a more amount of yolk, there will be an incomplete cleavage, and there is a less, otherwise a moderate amount of yolk, there will be a complete cleavage. So it is called as the holoblastic. So here, even in sea urchin also, the cleavage is holoblastic. Holoblastic means completely the egg will divide into two. But here in sea urchin, it is a radial cleavage. Okay, so it is a radial holoblastic cleavage. Coming to first cleavage, here first cleavage is a meridional. Okay, first cleavage is a meridional. Here you can see in the picture, meridional cleavage which will start from animal pole and it will reach as the vegetal pole. Okay, and it will divide the zygote into completely into two cells, two equal cells, because it is a holoblastic cleavage. First cleavage is a meridional cleavage. Okay, next coming to the second cleavage. So here second cleavage also meridional, but it is perpendicular to the first cleavage. Second cleavage is also meridional. It will start from animal pole and it will reach as the vegetal pole. But it is perpendicular to the first cleavage. Therefore, after the second cleavage, we will have four cells. After the second cleavage, we will have the four cells. You can see here third cleavage. Okay, so here third cleavage is the equatorial cleavage. Okay, so this will pass through the equatorial line. Third cleavage is a equatorial. Okay, so therefore this is a equatorial perpendicular to the first two cleavage planes. Okay, so therefore here what will happen? It will separate the animal pole and the vegetal pole. We can see here it will separate the animal pole and the vegetal pole. It will pass through this. Okay, so that is the blue color. One is the animal pole and it is the yellowish reddish color is the vegetal pole, and it will separate the two cells. Okay, so now after the third cleavage you will have 8 cells. Okay, 4 cells are towards the animal pole and 4 cells are towards the vegetal pole. Okay, so this is the third cleavage. Next, coming to the fourth cleavage. Here, fourth cleavage is different in sea urchin. Fourth cleavage will be very different than the first three cleavage. So, here what will happen in the animal pole? Sorry. In the animal pole, you can see that. So here the fourth cleavage, it will separately at the animal pole and the vegetal pole. It will divide the animal pole cells into a mesomeres. Here it will be a mesomeres. Okay, so equally. So to the animal pole separate the equatorial division will takes place. Okay, that is the fourth cleavage. Same the division will takes place at the vegetal pole also. So here what will happen? At the animal pole, it will divide all the 4 cells into equal cells. Those cells are called as the mesomeres. But what will happen towards the vegetal pole? So here towards the vegetal pole, it will divide unequally. It will divide unequally. Therefore, towards the vegetal pole, the 4 cells what are there on the after the third cleavage, at the fourth cleavage, these 4 cells will divide into 4 smaller cells and 4 larger cells. So these are called as the micromeres. Okay, so here you can see that in the picture at the lower side. So here you can see the lower micromeres and upper macromeres. Therefore, after the fourth cleavage in sea urchin, we can see 3 types of cells. Those are micromeres, macromeres and the mesomeres. Mesomeres are moderate in size, macromeres are a bit larger than that. Micromeres are smaller cells. Okay, so this is after the fourth cleavage. Next what will happen? So there will be a cleavage will be a separate at the animal pole and at the vegetal pole. Okay, so which cells will divide at the animal pole? It will be labeled as AN1 and AN2. AN is nothing but the animal pole, and towards the vegetal pole it will be labeled as the Veg 1, VEG1 and VEG2. Okay, so again, so AN1 and AN2 will be derived from the animal pole, and VG1 and VG2 will be derived from the vegetal pole. Okay, so afterwards the simultaneously cleavage will take place when it will reach us 128 cells. This is the cluster of cells. Then it will start with formation of blastula in sea urchin. So here after the 128 cells, so blastulation will start. Okay, that is called as the formation of blastula. So how the blastula will be formed? So here what will happen? The cells, they will push themselves towards the periphery, and at the center it will start to formation of the cavity. Okay, so here when it is a 128 cells, so all the cells will be a same size. Initially I said that there will be 3 types of cells like micromeres, macromeres and mesomeres. But once it will reaches 128 cells and once it will start to formation of blastula, all the cells will be a same size cells. By the time the cells will, what will happen? No, micromeres will slow down their division and macromeres will keep on dividing. Therefore, all the cells will become equal in size. Okay, so here At the center, it will be forming. Let me touch you. Yeah, here you can see that the cells will push themselves towards the periphery. At the center, it will form the cavity, and this is the proteinous fluid will be filled at the center. And at the periphery, you can see that only one layer of cells will be present. Only one layer of cells. Yes, here you can see that. So those cells are called as the blastula. Therefore, this is the blastula of sea urchin. Now, just I will tell the structure of blastula of sea urchin. Blastula of sea urchin is a sphere-shaped, and centrally it has got the large cavity which is blastocyst, and it will be surrounded by the same side cells called the blastoderm. Here what will happen during the blastula stage, some of the cells will start to develop the cilia. So cilia will be formed at the cells, okay, that is the blastoderms, okay. So here all the cells will not develop the cilia, but few blastoderms will develop the cilia. Here you can see that in the picture, okay. So here it is a blastocyst, and this will form the future ectoderm, and here it will give rise to future endoderm, and this is the future mesoderm, okay. So here you can see that basal lamina, and here what you are observing, this is the cilia, okay. So this is the cilia, and this here you can see that it is the hyaline layer. This is the vegetal pole, and this is the animal pole. You can see one changes here. Towards the vegetal pole, the cells has become flattened here. Okay, so this will form vegetal plate later during the gastrulation. So this will form the vegetal plate. Now only it has become flattened, cells have become flattened. Therefore now it is called as the vegetal plate. So here you can see the once again here you can see the hyaline layer. These are the cells. Cells has developed this cilia, and this is the future ectoderm, endoderm, and the mesoderm. Here you can see the large blastocyst. Okay, so this is the animal pole. So this is the blastulation in sea urchin. One thing you should remember, here in sea urchin, in the blastula stage itself, it will hatches from the egg and it will come out. Because it has got cilia, therefore it can freely swim in water. Okay, so with the help of cilia, it can freely swim in water. Therefore, the in sea urchin only in the blastula stage it will come out from the egg. But if you see in frog, otherwise chick, no, in frog only the tadpole stage it will come out from the egg. But if it will see in chick, so there will be a complete development will takes place. Chicklet only will, that will be a miniature of the adult, it will come out because so main reason is it has got the more amount of yolk which will support complete development of the embryo. In frog, if you will see, it is a mesolesical moderate amount of yolk. Okay, so therefore it will not support to complete development of the frog. Again, if you will see in sea urchin, it is a microlesical, really small amount of yolk. Therefore, it cannot support the complete development of the embryo. Therefore, in blastula stage, it will come out from the egg and it will grow into a larva. Okay, so this is about blastulation in sea urchin. Next class we will see the blastulation in frog. Okay, so then we have to compare the blastulation in all the three. Thank you, viewers. Next class we will see blastulation in frog. Okay, as well as followed by blastulation in chick. And also finally we have to compare all the three. Thank you, viewers.